So this time our topic of discussion is alcoholic liver disease. As we know, alcohol it affects the liver directly besides affecting other system. Now three things can happen if we drink excessively. These are alcoholic fatty change, which is known as hepatocellular steatosis, then alcoholic hepatitis, and finally the liver cirrhosis or alcoholic cirrhosis. So these proceed in a sequential manner. So today we are going to discuss first alcoholic fatty change and alcoholic hepatitis. We will leave the liver cirrhosis for our next lesson. So first, alcoholic fatty change. Now what happens, as we have discussed the basic morphology of liver, it is the largest gland. Then it is 1400 to 1600 gram weight. Then of course there are right lobe, left lobe, we know from anatomy, and of course this has been discussed in the general pathology. Now what are the changes grossly in case of alcoholic liver disease is that the liver, the margins become rounded and the liver is enlarged. Of course the margins become rounded and the liver gets enlarged. The margins are originally sharp, they get rounded, and on cut section what are the features is that there will be yellow greasy secretions from the liver, and the liver is firm with a smooth and glistening capsule. So these are the grossly what we can see in case of an alcoholic fatty change. Now after gross, let's discuss the microscopic findings. What happens is that initially there will be some microvesicle formation. These are all fat vesicles, microvesicle formation within the liver cells, microvesicle formation. These all occurs in the hepatocyte cytoplasm, microvesicle formation. Then followed by there will be a macrovesicle formation. As the disease goes on progressing, there will be macrovesicle formation. Now, as we know from general physics that two drops of water, when they combine, they form a large drop. Similarly, macrovesicle and a microvesicle on combining, or a micro combination, or a macrovesicular combination, all these can lead to a formation of a fat cyst. It's a large one, a fat cyst. So these in brief are the gross and the microscopic features. So next our topic of discussion is the alcoholic hepatitis. Now in sequential progress, next comes alcoholic hepatitis. Hepatitis has got various other etiological causes, like in viral hepatitis is there, and then but today we are going to discuss alcoholic hepatitis, of course. Now alcoholic hepatitis, what happens, it develops acutely, means it is a sudden phenomenon. After usually a bout of heavy drinking it occurs, and this is a forerunner of liver cirrhosis. So now what are the microscopic findings here? So there is alcohol leads to injury, so there will be necrosis, sudden acute phase, and it will lead to necrosis. Now what happens in case of necrosis is that there will be a ballooning degeneration of the hepatocytes. As we know how a balloon looks like, the hepatocytes will get transformed into a balloon-like cell. This all occurs due to the accumulation of fat, water, and protein. Okay. Now there's a special kind of thing which happens here is the Mallory body formation. The Mallory body formation. Now what are these Mallory bodies? The very important one. These are eosinophilic, then intracytoplasmic inclusions. Now remember that these Mallory bodies will occur only in the balloon hepatocytes. Eosinophilic, then these are intracytoplasmic inclusions. These are perinuclear, means surrounding the nucleus. Perinuclear, then of course found within the Solent and below hepatocyte. Now, what are these? How this come? How this eosinophilic perinuclear halo develops? These are phosphorylated cytokeratins, pre-keratin filaments, and heat shock proteins. So these three, and there are other also. These three leads to the formation of these eosinophilic intracytoplasmic perinuclear halo formation. This is the Mallory body. Now, Mallory body, we can visualize it with a connective tissue staining like Mason's trichome, then chromophobe aniline blue. So these are the two important stains. Or also there is a immunoperoxidase method. Now, these Mallory bodies are suggestive of alcoholic hepatitis, but not pathognomonic. Suggestive but not pathognomonic. What it means that Mallory bodies can be found in other diseases also, like in case of Indian childhood cirrhosis, biliary cirrhosis, and cholestatic syndrome, Wilson's disease, and in case of hepatocellular carcinoma, of course. So these are the conditions where Mallory bodies can be found also. So Mallory body is suggestive of hepatocellular, means alcoholic hepatitis, but not pathognomonic. That you must remember that. Now next, there is an inflammatory response. Now as there is a swelling, necrosis, Mallory body formation, all the mess going on there, it leads to inflammatory infiltrate. Now all of us know what kind of inflammatory infiltrate, what infiltrates are. These are neutrophils, plasma cells, then lymphocytes, all these cells which participate in our immunity. Then in a more extensive necrosis, it can lead to fibrosis, means it involves the entire lobule. Okay.